Hello, everybody. Welcome to Ms. Maguire online video lecture. This is introductory to human anatomy and physiology class, and today's lecture is about blood. So let's go ahead and begin. So we'll talk about function of blood first. So first is transportation. So your blood transport oxygen, carbon dioxide that's considered to be a waste product. Now, where exactly it transported and to what organs? So you know, where do you get blood rich in oxygen, right? Those are blood vessels that pick up oxygen in your lungs, right? So from lungs, your blood picks up oxygen and carries this oxygen to your cells. And your organs are made of cells. So when organs receive oxygen delivered by blood from lungs, we say cells receive oxygen. So oxygen moves from lungs to cells, and then cells, through metabolic processes, produce waste product, carbon dioxide. So this carbon dioxide from cells, organs, need to be moved back to the lungs so you can exhale it and get rid of this waste product, right? So all of this done by your blood. Your blood also transport nutrient, glucose, amino acid, fat. Now, where do you get these nutrients? When you eat your food, you digest it. So you break it down into nutrients, and those nutrients, such as sugar, amino acid, proteins, you absorb them in your blood, and your blood takes it to your organs. And other molecules, such as cholesterol, urea, and hormones. Now, your blood carry endocrine hormones from your glands, endocrine glands, to their target organs. So that's first function, transport. Another transport is regulation, maintaining body temperature or heat transport. Now, when it's hot outside, then blood moves to surface and heat radiates off the face, like when you are flushed. So you know when you're hot or when you're running or you do some physical activities, your face get pink. That happened because the blood rushes to the surface of your body, your blood vessels dilate, and extra heat is radiated from your skin to the environment. So that cools you down. In cold weather, blood vessels move your blood inwards to prevent heat loss, that when you get paled. So when temperature outside is cold, your blood vessels constrict and less blood flows to your skin. So you're losing less heat. Also, your blood regulates pH. In your blood, you have what is called buffer system. An important part of this buffer system is bicarbonate. All right, so this is how you keep your pH balanced. Another function is defense, fight off pathogens. You have in your blood, not only you have your red blood cells, because transport is due to red blood cells, and plasma, for example, carbon dioxide is transferred in plasma, oxygen is mostly transferred inside red blood cells, but you also have white blood cells and those that fight pathogens. Now, what are pathogens? Those are agents that cause infections, such as bacteria, viruses, and parasites. So your white blood cells break down pathogens, they eating them and breaking them down into pieces that then you either get rid of them or you can recycle them. Okay, so that's function of blood. Now, what is blood made of? Now, first, blood is a tissue. It's a connective tissue, fluid connective tissue. And if you draw blood from a patient, of course, you know, you can draw from a superficial capillaries, or you can draw from veins, and put it in a test tube, and then you centrifuge it. Then blood will be separated into several compounds. because when we spin blood sample, heavier components, they settle. So the heavier components will be on the bottom. Then we will have this buffy coat, and the lightest part of blood will be on the top. So on the bottom, you will have erythrocytes, so it's your red blood cells, and they will be about 45% of the volume. This buffy coating gonna be a formed element. So this will be white blood cells and platelets. And on the top, you will have plasma that is about 55% of whole blood. So now let's look at plasma. So plasma is mostly water. It's 91% water and 9% of ions like potassium, sodium, molecules such as proteins, glucose, and fat, urea, that is nitrogenous waste, and bicarbonate. This is a part of our buffer system. So those proteins of the plasma, there is 3 main proteins. All these proteins produced by liver, right? So liver make proteins of your blood. Now, what are those proteins? So first is albumin. This is 60% of all plasma proteins, and it's important in osmotic pressure. So it keeps water in blood and away from tissue. Now, if you want to look at the albumin, like how albumin would look like, if you have a raw chicken egg, those whites that you have, it's mostly albumin. So another group of proteins are globulin. This is about 36% of plasma proteins, and they are important in transport of vitamin, lipids, and hormones. And about 4% of plasma proteins are fibrinogen. Those are protein important in the blood clotting. So when you have a cut or some other injuries that cause bleeding, you want the bleeding stop, right? So, and the first process in hemostasis or stopping bleeding is blood clotting, right? So it will be platelet plaque. So you will have platelets that form a temporary plaque, and then you will have this fibrinogen that will seal blood vessels for good. Right, so that's our 3 types of plasma proteins: albumin, globulin, and fibrinogen, and again, all produced by liver. So red blood cells, or RBCs, or erythrocytes. Cytes means cell, and erythra means red. So those are biconcave disc. So if you see over here, right, so that's a disc, and it has like a little like this concave part of it, right? So like a little depression inside the disc. That's what this means, biconcave disc. Now, each RBC is packed with protein called hemoglobin, or HB. So on this picture, you see the structure of hemoglobin. Of course, this is a cartoon picture, right? And this is a chemical structure of heme group, right? But we will look at hemoglobin in a minute. So most of the volume of red blood cells are actually hemoglobin. So in this picture, it's a 400x magnification. You can see a capillary and red blood cells inside the capillaries, and you see the red blood cells almost the same as diameter of capillaries. And red blood cells, by the way, one of the smallest cells in your body. So we have about 250 million hemoglobin in each RBCs. So what is this hemoglobin is made of? So you can see this diagram here, and all this blue stuff shown here in light and dark blue. This is actually protein. Now, protein is called globin. So we have alpha, 2 alpha chains, and 2 beta chains. So the alpha chain shown in the dark blue, and beta showed in light blue. So that's a protein. This is a globular protein. Remember, like protein are made of amino acids that have primary, secondary, tertiary structure, right? So this is just functional protein. So in each hemoglobin, we have 4 globins. Then you see this yellow part, that's a heme. So we have 4 molecules of heme, and this green part, so over here green, this is the iron. So we also have 4. So that's so the structure of hemoglobin. So this is a whole molecule that's very important in transportation of oxygen, made of 4 proteins, 4 globin, 4 heme group, and 4 iron. Now globin. So iron, let's start with iron. So iron function is to carry oxygen. So actually oxygen binds to iron right here. Globin can carry carbon dioxide. However, most of the carbon dioxide carried by plasma, but some carbon dioxide carried by globin. Now the heme group, that's what give blood red color. What do we need heme group for? Well, the heme group actually attaches iron, and iron attaches oxygen, right? So that's a structure of one hemoglobin. Now, so if you take one red blood cells, inside that one cell, you will have 250 million of hemoglobin molecules. Now, each molecule of hemoglobin attaches 4 molecules of oxygen, right? So if you multiply 250 molecules of hemoglobin by 4, because each hemoglobin carries 2 oxygen molecules, that means inside one red blood cell, you have 1 billion oxygen molecules. So it can carry 1 billion oxygen molecules. Sometimes it carry less, but the maximum it can carry 1 billion molecules of oxygen in one red blood cell. So transport of oxygen. Now, when hemoglobin attaches to oxygen, so you see, so here's your red blood cell. So inside we have this molecules of hemoglobin. So oxygen binds to hemoglobin, then we call it oxyhemoglobin. So when oxygen binds to hemoglobin, and this one is ruby red color in blood. Now, so why do you need bind oxygen to hemoglobin? Because this is how you carry it from lungs to your tissue. When you reach your tissue, when your blood reaches your tissue, oxygen is released to those tissue cells. When hemoglobin releases oxygen, we call it deoxyhemoglobin. So when hemoglobin releases its oxygen, and this one is dark red color. And under skin, it appears blue. Now, you don't have, of course, blue blood, right? Your blood is always red. But in your arteries, in most of your arteries, blood is ruby red, and most in your veins is dark red color, and under skin, it just looks blue, but it's always red color. Your blood also transfer carbon dioxide. So most common way of transportation of carbon dioxide in the form of bicarbonate, so HCO3 minus. So carbon dioxide converted into bicarbonate, and bicarbonate moved to plasma. So most of the carbon dioxide carried by plasma, not by red blood cells. So here's your reaction. Carbon dioxide reacts with water, became a carbonic acid, and then carbonic acid dissociates, so it splits into hydrogen ion and bicarbonate ion. And this has happened in plasma. But some hemoglobin can carry oxygen as well. And where is this hemoglobin? Inside red blood cells. Right, so this is a small amount. But when hemoglobin carry carbon dioxide, we call it carbaminohemoglobin. So this is the second common way of transporting carbon dioxide when carbon dioxide binds to globin. Carb, it means CO2, amino acid, and hemoglobin, carbaminohemoglobin. So that's what this word stands for. Because what is globin? Globin is protein, so it's made of amino acid. That's why we have carb, amino, hemoglobin. So we have carbon dioxide, we have protein globin that made of amino acid, and globin in this word means hemoglobin. Okay, so where red blood cells are made. So production of red blood cells take place in the red bone marrow. Now, where this red bone marrow is located? Well, because it's a bone marrow, that means it's somewhere in your bone, right? So the red bone marrow in adults located in a spongy bone. So it will be in the flat bones or in epiphyses of long bones. Produce an average of 2 million RBCs every second. And this production controlled and rely on a special hormone called erythropoietin. Now, this erythropoietin, or EPO, produced by kidney. EPO goes to red bone marrow to stimulate production of red blood cells. Right, so your red blood cells, they are short-lived. They live in average 4 months, right? So red blood cells live only 4 months. And every 4 months, you need new red blood cells, right? That's why every second you synthesize 2 million of those red blood cells inside your bone marrow, red bone marrow, and you need a special hormone produced by your kidney, EPO, to control and stimulate this process. So what happens if you don't have enough oxygen in your blood? So low oxygen, called hypoxia. So hypoxia is low oxygen blood level. So when in this blood flow to the kidney, right? So kidney actually is very vascular organ. So lots of blood flow to the kidney, and then kidney filters the blood, and filter blood return back from the kidney. But in the process of doing it, when blood flows to the kidney and flows back, you have a special receptors in your kidney that sense low oxygen level. So when oxygen level is low in blood, kidney release more erythropoietin. So kidney increases production of erythropoietin. So erythropoietin then released to the blood, and blood carried to the spongy bone. So this is a epiphysis of a long bone where you have red bone marrow and you have stem cells that increase red blood cell production. So oxygen level returns back to normal, right? And that's back, so we're back to normal over here. So it's go low again, we start this process all over. So this is negative feedback, right? Oxygen level is low, and our result is normal oxygen level. So this is the example of negative feedback. So we have our stimulus, low oxygen level. We have kidney that senses this low level. Then it's also it also release erythropoietin. Then we have stem cells in the red bone marrow that are effectors. They response would be increase synthesis of red blood cells and result normal oxygen level. So here's our RBC destructions. So RBCs are packed with hemoglobin. So RBCs then have no room for nucleus and organelles. So if you have a cell and most of this volume is hemoglobin, then you don't have enough room for those huge organelles like nucleus, for example. And because RBCs do not have nucleus, or other important organelle like mitochondria, then RBCs are unable to repair itself. So average life is about 120 days, relatively short. And 2 million RBCs destroyed every second and 2 million RBC produced every second. So that keep your red blood cells count constant, right? So 2 million produced and 2 million destroyed every second. So that keep your body in balance. So you don't lose your RBCs. So what is this RBC made of? So we already said that RBC, red blood cells are cells. They're not really normal cells because they're missing important organelles like nucleus, mitochondria. So we call them formed elements. And mostly they're made of hemoglobin. And hemoglobin, we just discussed it, it's made of 3 parts: globin, protein subunits, heme, and this is a pigment, and iron. So now if you destroy 2 million red blood cells every second, so what's going to happen with all these parts? So because this globin protein broken down into amino acid and reused for protein synthesis. Heme broken down to pigment called bilirubin. Bilirubin sends to liver and it's transported to digestive system and excreted with feces. So color of feces is a removal of bilirubin. So you notice that doesn't matter what color food you eat, your feces are always yellow-brown, right? So this yellow-brown color is because of the destruction of heme. Right? So your blood cells die, this heme converted into pigment that is yellow-brown in color. And we have iron. Iron is returned to bone marrow also for use or stored in liver. So we reuse globin, we reuse iron, right? And heme, this pigment is, it's actually used to make a bile. So, and the rest of it is excreted with feces. So, so here's our diagram of what your body does with red blood cells. And because most of it hemoglobin, so what your body does with hemoglobin. So globin protein subunits broken down to amino acid, and amino acid used to produce new proteins. Heme broken down to bilirubin goes to liver, released into digestive system, and removed in feces. And iron sent to red bone marrow and reuse it, so we recycle it to produce new red blood cells or store in the liver for future use. Disorders of blood over here, jaundice. So jaundice is yellowing of the skin and whites of the eyes. So you see this is a normal hand and this is jaundice patient, and this is the eye of a patient with jaundice. So why the skin and whites of the eyes became yellow? It's because of inability of liver to excrete heme byproduct. So if you go back over here, right, so this heme is broken into bilirubin and your liver need to release it into digestive system and it's need to be removed in feces. If your liver failed to do it, then this bilirubin gets into blood and blood carries it through your body, and your skin and your eyes have this yellow color. So here's some cool fact. Changes in the skin color after bruising is because of chemical breakdown of hemoglobin in the site of injury. So you all know how your bruises change with time, but what is bruise? Bruise is actually the small hematoma. So bruise happens when small capillaries are ruptured and blood leaked out under your skin, right? So that's a blood under your skin, and blood has plasma, of course, and has red blood cells. Red blood cells are full of hemoglobin. Hemoglobin is breaking into biliverdin first and then bilirubin. And all of these heme and biliverdin and bilirubin, those are pigments. So, and this is how their color changes when bruises progress. So if it's a red, swollen, and tendon, that's a bruise of 0 to 2 days, blue or purple, 2 to 5 days, green. So here, see this green color, is 5 to 7 days, and yellow, 7 to 10 days, then brown, 10 to 14 days, and then 2 to 4 weeks. We should have no evidence of bruising because it's all your body took care of it, so you recycled and broke down this blood under the skin. Another disorder of blood is anemia. So anemia is actually low oxygen level in the blood. So we have different type of anemia. One of them may be because of the low count of RBCs. So symptoms include pale skin, fatigue, short of breath, and chills. So here's one type of anemia called sickle cell anemia, when cells are sickle cell shaped. So this is normal red blood cells, and this is sickle celled. So this defect in a shape of red blood cells is inheritary. So this is genetic disorder. This is genetic disease. When the gene that is responsible for hemoglobin synthesis has mutation, and then cells are formed with a wrong shape. So this leads to hemolysis or rupturing of red blood cells, causes blockage of blood vessels, and treatment include blood transfusion. So this is one type of anemia, but we have several anemia. Here, for example, renal anemia. When kidney disease causes decrease in erythropoietin level, EPO, and we know that EPO is needed to stimulate production of red blood cells. So if a kidney does not synthesize enough of this hormone, then low EPO decreases production of red blood cells, and treatment is injection of erythropoietin. Blood loss can also lead to anemia. Blood loss as a result of accident of hemorrhage. Hemorrhage is bleeding, treatment, blood transfusion. And this is iron deficiency anemia. It's actually the most common type of anemia when we have low level of iron, so body can produce RBCs, and treatment increase iron intake in a dietary or supplements. Okay, so now Buffy coating. So let's just go back for a second because I want to show you what we talking about here, right? So if you know, if we centrifuge blood, so where is that? Right here, right? So if you have a whole blood, it's made of red blood cells, Buffy coating, and plasma. And that's why we talk about plasma first. Remember, we talk about plasma, so it's mostly water, then it has iron and urea, bicarbonate, and proteins, glucose, fat. So here's our 3 proteins of plasma. So we talk about this part, plasma. Then these red blood cells, those so those are we talk about over here. So we talk about function of red blood cell structure, where they formed, right? So how they destroyed. And so that's red blood cells, right over here, or erythrocytes. But now we're going to talk about this Buffy coating that include red blood cells and platelets. So this is over here. Now we talk about, okay. So what is this Buffy coding? Those are red blood cells and platelets. So white blood cells, WBC, also known as leukocytes, function is defense or immunity of the body, and platelets essential for blood clotting. So when we're looking at this white blood cells, so platelets actually those are fragments of megakaryocytes. So you have a special cells, megakaryocytes, and fragments of them form platelets. But when we're talking about white blood cells that are important in defense, we have 5 different types of white blood cells. So here's our classification. So neutrophils, 50 to 70%, lymphocytes, 25-35%, monocytes, 2 to 8%, eosinophils, 1 to 4, and basophils, 0.5 to 1%. So the mnemonic over here, you see this first letter, NLMEB. So mnemonic is never let monkey eat bananas. Right, so this way you can put them in the order of the more frequency, from more frequency to less frequencies. Neutrophils, their function is to phagocytize pathogens and cellular debris. So they are so this is macrophage. Monocytes are macrophages. So what macrophages do, they engulf and destroy pathogens. Neutrophils called microphages. So they also phages, but they're smaller than monocytes. So eosinophils and the basophils, all of this actually has some of phagocytic properties. So neutrophils fight bacteria and cellular debris. Eosinophils use granule content to digest large pathogens such as worms and reduce inflammation. Basophils promote blood flow to injured tissue and inflammatory response. And those called granular leukocytes, because if you look inside the cytoplasm has this visible granules. And we have agranular leukocytes, so cytoplasm doesn't have visible granules, lymphocytes and monocytes. Lymphocytes are responsible for specific immunity, and it's we have 2 types of lymphocytes: B cells that produce antibodies and T cells that destroy cancer and virus-infected cells. And monocytes become macrophages that phagocytize pathogens and cellular debris, like this. The same, similar to this one, but the neutrophils are granulocytes, monocytes are agranulocytes. They have different shape, they have different structure. Neutrophils has visible granules and monocytes don't, but there is some similarities in function, right? Neutrophils phagocytize pathogens, and monocytes phagocytize pathogens and cellular debris. And the platelets, known as thrombocytes, and function in blood clotting. So when you look at the slide of blood smear, you'll see this is our red blood cells, and those like a smaller fragments of cells are platelets, and this fluid in between is plasma. So here's some more disorders of blood, like leukemia. It's a form of cancer, and it affects white blood cells, and thrombocytopenia, abnormally low concentration of platelets, and it will affect the blood clotting ability of a patient. Okay, and this is where I'm gonna stop this lecture. So the next lecture will be about blood typing, right? So, okay, thank you for watching, and I hope it was helpful.